Hello students, I am Dr. Bhumija Sharma. I am here to present the class on urea cycle. This is a cycle which is also known as Krebs-Henselt cycle. Urea cycle means we are converting the formation of urea from ammonia. Ammonia, which is very highly toxic for our body, so it has to be removed from our body immediately as soon as possible. But being ammonia very toxic cannot be holded by the cells, so it is converted into a milder form where it can be converted, it can be stored for a little while and is a non-toxic product. So the urea is being produced. Now, this urea cycle is the first metabolic pathway. It is to be elucidated that the cycle is also known as Krebs-Henselt urea cycle. The ornithine is the first member of this reaction. It is also known as the ornithine cycle. So, urea cycles, the names are Krebs-Henselt cycle or ornithine cycle. It is synthesized in liver and transported to kidney for excretion in urine. Now, this urea is formed from the ammonia, and the main thing is ammonia has to be detoxified. The ammonia detoxification takes place with the help of glutamine. The ammonia is trapped in the glutamine. It is reduced production, ammonia trapping, and urea synthesis. These are the steps of the removal of ammonia. This is a complete structure of urea cycle. How ammonia is trapped with the help of carbon dioxide. First product formed is carbonyl phosphate, which is reacting with ornithine to form citrulline. Then arginosuccinate is formed, then arginine, and then again the cycle is turned over to the ornithine formation. Now we will discuss this cycle step by step. In the first enzyme which is required for the formation of carbonyl phosphate synthesis is CPS1. There are two kinds of CPS enzymes, CPS1 and CPS2. One is in the mitochondria and the second one is in the cytosol. The mitochondrial enzyme is useful in the urea cycle, but the cytosol CPS2 enzyme is useful for the production of pyrimidine. These are the two different same enzyme but at two different places and locations, so their utilization is different. CPS1 is dependent on N-acetyl glutamate, which is also known as NAG. CPS2, it is totally independent of NAG. CPS1 is also activated by the ammonia concentration. Ammonia has to be concentrated and formed urea and it has to be removed from the body. All these process of the urea cycle occurs in the liver. The formation of urea takes place and then it has to be given out to the kidneys where it will be excreted out. Now, the step one, the ammonia, which is coming, a toxic product coming with the help with the carbon dioxide and water, it is forming the first molecule of this cycle, which is carbonyl phosphate. The enzyme is carbonyl phosphate synthetase. This is one enzyme in the mitochondria. The carbonyl phosphate is being formed in the mitochondria. Two steps are done, and then the rest is cycle is in the cytosol. This carbonyl phosphate again with ornithine will form the product which is known as citrulline. Now ornithine and citrulline, both of these are basic amino acids, but they are not found anywhere in the protein structure. Please remember students, these are the non-protein structure amino acids because they don't have any code languages also with them. These are ornithine and citrulline, basic amino acids not found in proteinaceous structure. After the formation of citrulline, the step 2, citrulline is converted to arginosuccinate with the help of the enzyme arginosuccinate synthetase. Here the ATP formation is being utilized and it is converted to AMP. That means about 2 ATPs are utilized. Equivalency is around 2 ATPs. Aspartate here is coming along with citrulline and it's forming arginosuccinate. This aspartate is a biwave from the TCA cycle. This is a product which is coming from TCA cycle and the arginosuccinate is being formed. After this, there will be next step in which arginosuccinate is being splitted to form arginine and fumarate. Once again, this is a step which is being related to TCA cycle. Here, this fumarate will go into the TCA cycle to form the aspartate, and this is a connection between a urea cycle and the TCA cycle. The arginine which is being formed over here, this will be converted to the urea directly. It is a precursor of urea, you can say. Arginine, last step is the cycle step 5, where arginine is converted back to ornithine and urea is product as a product is being formed, and the cycle of urea is completed over here. Ornithine again will be utilized with the help of CPS to form the citrulline. This completes your turnover of the urea cycle. This fumarate, which I have told you again, is connected to the TCA cycle and converting back into aspartate, which will connect with citrulline to form arginosuccinate. So this is a linkage between a urea and TCA cycle. This is an overall picture which you can see the what is happening in the cells during the formation of urea. This is being converted in CPS and citrulline in the mitochondria. All other these reactions are being done in cytosols, and this again malate and oxaloacetate mitochondrial reactions are being seen. This is an overall picture by which the urea is formed. Now, when the urea has been formed, it will be transported to the kidney for the excretion. Now, suppose if any of these enzymes are being lost or are be deficient, then there will be increase, not no production of urea, and ammonia will be accumulated in the body's tissues and the cell, which will create the disorders of the ammonia, which we can continue in the next classes. Energetics of urea cycle. The overall reaction is summarized as ammonia, carbon dioxide, aspartate, and it will form urea and the fumarate. Urea cycle, in this 2 ATPs are utilized in first reaction. Another ATP is converted to AMP, which is equivalent to 2 ATPs. So 4 high energy phosphate bonds are utilized. However, fumarate formed in the fourth step may be converted into malate. This is a TCA linkage. Next is the part of the urea disorders, which we can continue in the next class. Malate oxidized to the oxaloacetate will produce NADH equivalent to which is equivalent to 2.5 ATP. No net charge expenditure is in and only 1.5 high energy phosphate is produced. The urea cycle and TCA cycle are interlinked, and so it is called as urea bicycle. Regulations of the urea cycle is due to the course regulation. During the starvation, the activity of urea cycle enzymes is elevated to meet the increased rate of protein catabolism. The fine regulation is done by the majority enzymes of CPS I and acetyl glutamate, which will stimulate this reaction. It is formed from glutamate and acetyl coenzyme A. Arginine is also an activator of NAG, and acetyl glutamate. Thank you, all of you. We will continue with the urea disorders in the next classes.